Welcome everybody to our second February meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. Appreciate you coming to hear our two speakers and talk this afternoon. As always, I need to remind you that this meeting is live streamed on YouTube and the videos are recorded and made available on YouTube after the meetings. As always, Reinhardt and myself are available to take your questions and suggestions. Reinhardt wasn't able to make it today. I believe he's on study section. So he's absent today. He'll be back next week. Jim Sluka continues to help us out and be one of the leads of the group. And Lorenzo Veschini has been stepping in. People have had a lot of communication with him. Please feel free to suggest ideas to him. As always, please help us try to let people know about what we're doing. We'd especially appreciate updates for the IMAG MSM wiki page. We've gotten some suggestions from Grace that we need to do more in terms of updating our web presence. And so your help would be appreciated. And always distributing the YouTube videos for the meetings would also be appreciated. And we do have that Twitter feed, twitter.com MSMviral. Please help us use it. Feel free to contact us about how to use that. Jim Sluka has created that and made it available for people to use. Are there any particular announcements that people would like to make before we start the main meeting? I was going to mention that tomorrow will be the first meeting of the new Building Digital Twins subgroup at 11 a.m. Eastern Standard Time. If people are not invited to that meeting and would like to be, please email Reinhardt and Bruce or Lorenzo for the links for that meeting. Everyone is welcome. Any other announcements? I know Grace circulated an announcement about collaborative workshop on collaborative science that she's organizing at NIH, and that might be of interest to people. So coming up, we really need your help for speakers. Please do send suggestions. Next week, we so far had Giuliano Gianlupi scheduled to talk about some modeling that he's been doing. We have a speaker the following week also. There's nothing wrong with only having one speaker in a week, but we certainly can accommodate two. We definitely would appreciate your suggestions starting next week and going forward. I know Lorenzo's been doing a lot of work trying to reach out and secure speakers. Ideas can be very lateral. A modeler you want to hear, a modeler in your group that you want us to hear, experimentalists, clinicians, industry people. We've had journalists speak. We've had people from all sorts of different fields speak, veterinary medicine. So please do help us come up with ideas. I think there's a lot of the world of infection and immune response that we haven't explored yet in the working group, and your ideas about interesting speakers would be much appreciated. Of course, we also have people talking about model sharing, standardization. So almost no topic is too offbeat as long as it can help us understand how to address these complex phenomena. So please do send your suggestions. Lorenzo, do you want to make any update on where we are in terms of meeting scheduling?
Well, as you mentioned, we have a few speakers planned up, but obviously people that want to talk or have some suggestion for invitation are more than welcome, because we can for sure accommodate We have a lot of slots coming up April and March also. So please step forward if you want to talk.
And as always, your graduate students and postdocs are welcome. So it doesn't have to be senior people speaking. And if you've spoken before, you can speak again. And also reports from the subgroups. In particular, I know, Veronica, you've been involved in a very active subgroup. If you'd like to use some time to open up those discussions here, that would be fine. And others are welcome to do the same. I know Yi, you've been thinking about some things that might be appropriate to discuss in this meeting as well. It doesn't have to be a formal presentation. We've had meetings which were primarily discussion, and that's absolutely okay. All right. So both of our speakers have agreed to stay on for discussion afterwards. Both of our speakers have spoken to us before. Both had plenty to say, so we're glad to have them back. I'd like to start with Kevin Janes, who gave a wonderful talk early in the series and is coming back to talk about stress testing complete kinetic models of individualized enteroviral infection. And I will turn it over to you.
Jim, can I get a thumbs up that you can see my slide? Okay, I see the head nod. Terrific. Thank you very much, Jim and Lorenzo, for the invitation back. As Jim said, I spoke here, I think within the past year or so. And the last time I did, I received plenty of follow-up emails and things. It was terrific. I just wanted to warn in advance, if you do that now, I might be especially slow in responding because since the last time I presented, I've become director of graduate admissions in my program, and it's the graduate recruiting season. I'm pretty overwhelmed with the extensive correspondence these days, but I will be sticking around afterwards. So if you have questions right in the moment, probably the best time is to talk with me about them at the end. My talk today is going to be a bit of an experiment on its own, a social experiment, one in openness and transparency. And so what I wanted to try to do is give a grant brew. I've been at University of Virginia long enough as I'm not sure if that's just an UVA jargon or it's a universally known thing. But I wanted to speak with you about is a proposal that we're planning on submitting in about a month. That's pretty mature, but I'd like to get a sense from this audience of questions, concerns, or general level of excitement of some of the ideas that we have running around in our head or planned in my group. I know this meeting is broadcast and recorded and publicly available, but my thoughts are is that if the ideas that we're putting forth here are truly innovative, I should be able to share them widely. If you want to give it a try on these things, go for it. This stuff's really hard. So, okay, I get the virtual applause from Jim. Thank you. What we're trying to do in this application is leverage two publications that we put out in 2021, so pretty recent. And in fact, what I talked about was the cell systems. This was before it had been published, but the paper up in the upper left-hand side. And resubmit hopefully a competitive A1 application on deep mechanistic modeling for a family of RNA viruses called Enteroviruses, which includes the cardiotoxic virus called coxsackievirus B3, which is the original virus that we built some of these detailed mechanistic models for, but try to build off of those and expand into other areas of virology. As a little bit of a background, the modeling paper that we published last year is a mechanistic reaction differential equations model that abstracts, but with a relatively high level of detail, the viral life cycle for enteroviruses. And we built it in a modular framework broken down by the delivery of virions to the host cell plasma membrane, entry of the single-stranded RNA genome into the cytoplasm, and then conduits for that single-stranded RNA. So I can get my laser going here. giving rise to a translated product that yields mature proteins encoded by the viral genome that enable polymerase activity for further viral replication, structural proteins that encapsulate mature virions, as well as these enteroviral proteinases that are important for maturing the polypeptide encoded by the single-stranded RNA genome, but also go on and do collateral damage on host cells. Of particular interest to us were the way in which these would antagonize the antiviral interferon response that is mobilized by the host cells in response to the double-strand RNA intermediates that are a requirement for enteroviral replication. And if these are fully engaged, they can shut down the viral life cycle at a variety of points shown here. This model, and because enteroviruses overall have been studied for several decades, is pretty well constrained by the existing literature, our own experiments. Our estimate is 90+% of the parameters are taken directly from our own experiments or from literature values. But several of these negative feedbacks here, which are lumped parameters defining the interferon antiviral response that I described a moment ago, these are phenomenological parameters that are fitted qualitatively to the characteristics of the model. What I'm showing in the upper right-hand panel here is a global sensitivity analysis to assess which of these feedbacks are particularly sensitive to. And the take home here is that when we do a global analysis, we're not seeing effects that are all that much greater than the main effect of the single parameters, indicating to us that there's not a lot of emergent sensitivity of combinatorial parameters. And in fact, the paper, we do a lot of single parameter sensitivity analysis. And this is, I think, evidence that that's a good approximation for this model. When we take those 90% of literature or experiment-derived parameters and these fitted ones, we can achieve quite a good calibration to the kinetics of accumulating positive strand RNA genomes, as well as the template, the negative antigenome over time, the accumulation of viral proteins, as well as cleavage of host cell proteins, such as the initiation factor IF4G. That's a direct substrate of one of these enteroviral proteinases. And one of the things that we discovered as a result of looking at this model very deeply was a key role for an antiviral transducer called mitochondrial antiviral signaling protein, or MAVS, that transduces the double-strand RNA sensing to the mobilization of interferon-stimulated genes. And what we identified were multiple sites within the MAVS coding sequence that were themselves direct targets of the enteroviral proteinases. Interestingly, one of these, which had been largely overlooked up to this point, has a variant within the human population where some of us have the ability of this site to be cleaved. They have the glutamine here that can be cleaved by the enteroviral proteinases, but 50% of the alleles from individuals of East Asian ancestry have a glutamate allele in this position and are rendered incapable of getting cleaved by MAVS at this position. And what the paper points to is that the presence or absence of some of these alleles dictate the activation propensity of MAVS to form polymerized filaments on the mitochondrial surface and mobilize interferon response in cells when they're infected with enteroviruses such as coxsackievirus B3. So we went from a mechanistic model to some critical determinants at the molecular level that we could span out to human populations and differences, potential differences in susceptibility. The other paper that I mentioned at the beginning tries to make this mechanistic model generally useful and accessible to people who are not modelers. Shortly after we published the paper, we were invited to write a STAR protocols method on how to use the model. And rather than walk through the code, what we decided to do is recode it with a graphical interface with very details, a lot of ability to change parameters and do different simulations, but really trying to target the model to the virology community or the educational virology community to illustrate the power of computation and how you could deploy it in a virologic setting. One other thing that I wanted to say here is that I should, to a good extent, because of who my co-speaker was at this meeting last time I spoke, I was side to side with Rauman Sharif, who is a contributor to the biomodels database. I got talking with him afterwards, and we've since put the model in the biomodels database. So it's of course on GitHub, and we have the published papers, but we wanted to put it in a solid repository that's been around for a while to have a permanent place where people could find it. Let's dive into the overarching premise hypothesis of this proposal that I'll walk you through at a high level. And I'll say something a little bit provocative and then try to explain what I mean. If you've read any philosophy of science or taken any experimentation or scientific practices, we learned that we're supposed to falsify hypotheses, not prove them. And what I would like to put forth is that we should be doing more of that, or at the minimum, there's a good utility to doing this more with models themselves. Now, some might interject and say, well, we know this. I mean, it's a famous quote, incorrectly ascribed to Einstein, that all models are wrong, but some are useful. If you think about that, that's an affirmative statement. It's trying to find the places where the model is good, and that's different than what I'm talking about. Second, you might say, we use models to test hypotheses all the time. This is also a valid way to use modeling, but I would argue that's like the method of concurrent alternative hypotheses, where you're trying to find the good, you take away the good from the bad. And that's not really what I'm talking about. What I'm talking about is taking a model that you think is working and actively trying to break it, finding out where it yields. And the best parallels I can find for this are actually in computer science and software. If you have complicated code and you want to make sure if it's really ready for prime time, there's a phase that undergoes stress testing, where you take the model, you take a set of code and you try to challenge it adversarially to figure out how you can get it to malfunction. And some of that's just for robustness and hackers and things, but it's not uncommon for code to be so complicated that you don't know where the vulnerabilities are until you go to seek them out. And this is really what we're trying to bring to the table and the organizing principle for what I'll talk with you about today. Our hypothesis is that by attacking our models from this stress testing perspective, that what it will actually do is make the models better from the standpoint of generalization across different biological contexts, different viral host contexts. And I'd like to argue that that is a falsifiable hypothesis, because this could be wrong. We could be stress testing the model, and all that could do is make us feel worse about our model, make us even less confident about it. But instead, what I believe, what I would hypothesize, is that it can make it better in the long run. And the way that we're going to stress test this enteroviral model is by diving into some of these modular sets of reactions that I described in the overview, the level of viral delivery, the level of viral replication, and at the level of these feedbacks that I talked about just a moment ago. So first, let's talk about the viral delivery module. This is taking figure subpanels from the paper described before. Coxsackie-virus B3 has 2 viral receptors on the cell surface that bind to the virus, traffic it to tight junctions, and then give rise to endocytosis, escape, and then delivery into the cytoplasm. It's at that viral delivery where you see the greatest variation from one enteroviral species to the next. And this is a complication, but it's also an advantage, because we can exploit that property and take out the CDB3 delivery module and plug in a delivery module for another enterovirus. And now we have another model that's a reasonably good approximation for that other species. We did this for the very well characterized enterovirus poliovirus in about a week or 2. We could have a model that could simulate its properties, and it could already quite readily make predictions about what would be the time course of infection if cells, for example, were stimulated in a delayed fashion with exogenous interferon. For Coxsackie-virus B3, the delivery is slower, so you can add interferon up to about an hour and a half and still completely thwart the infection. You do the same thing in our poliovirus model, and the horse is already out of the barn, and adding the same amount of interferon at the same time point has no effect. So in principle, something that we go forth and test if it was what we wanted to focus on. We don't want to focus on that. In fact, we want to be a little bit more aggressive with our ideas. And we would like to broaden to another family of enteroviruses called rhinoviruses, common cold, things that infect your sinuses and things. And there's a longstanding observation that these rhinoviruses infect preferentially at a lower temperature than body temperature. So 33 degrees is more effective at infecting the host cells than at 37. And so what we're proposing in this aim is to swap out the delivery module, put in a rhinovirus module, and then propagate temperature as a complex systems level perturbation throughout the entire model using well-known relationships that describe how rate parameters and equilibrium binding affinities change when you tweak the temperature. And these 3 or 4 degrees Celsius changes are not that big, but when they're propagated system-wide, and they're propagated irregularly system-wide because each reaction has its own enthalpies or activation energy barriers, it starts to become a complex thing that we really need a model to figure out. And so we've made some progress. We built a model for human rhinovirus 2. As you might imagine, not all of these activation energies and enthalpies are known, but we can screen through realistic ranges that have been documented in the literature. And we can find parameter sets where we can start the model at 37, drop it to 33 degrees Celsius, and get about a 2-fold increase in infectivity, which very recently we have just been able to reproduce in the lab. So these are plaque assays of infected host cells at either 33 or 37, rev up the temperature, infectivity goes down. What's really cool is if we take the exact same set of parameters and plug them into our coxsackieviral model, we don't see the temperature change. And neither do we see the temperature. We see a temperature change, but not in the opposite direction when we shift from 33 to 37 for coxsackievirus. So I think what we're poised to do by now with in vitro experiments and also these computational models is dive in and figure out mechanistically what is the delivery module of human rhinovirus doing. This is the only thing that's been changed between these two modules, and why is that conferring this inverse temperature dependent sensitivity. All right, moving on, because I know I want to be cognizant of the time. The second aim is on viral replication. There's a whole lot of details that we have in the paper on this, but just as a synopsis, the way that viral replication occurs in all RNA viruses is that it requires the co-option of host cell membranes. And this is where the bulk of viral replication takes place, is on the surface or in a cavity of these membranes. And in the paper, we made a computational argument that the major advantage to that membrane co-option for enteroviruses is to rapidly accelerate the viral replication by moving chemical reactions from a 3D volume to a surface. The way that that's encoded currently is through a discontinuous step in the viral model where it propagates for a period of time. And then once the virus has accumulated enough copies of the 3D polymerase that's responsible for replicating the RNA, it jumps up to full-fledged viral surface concentrated replication rates. And this is what gives rise to those kinetics of positive strand and negative strand that I'd shown before. What's intriguing is that the model is not at all tolerant to many other alternative formalisms of transitioning from no viral replication organelle, no membranes, stolen replication centers, to the full-fledged replication when it all is occurring on the surface. If we put in a ramp, the model discrimination is poorer, and the fits are very uncertain when we fluctuate some of the parameters through. And it's drawn towards this because there's been a long-standing, now 30-year observation in the literature with poliovirus that if one can reconstitute poliovirus replication in a test tube ex vivo, with bits and pieces of cells providing parts of the membrane. And the authors in that early paper noted a 2 order of magnitude range in infectivity in this in vitro preparation that's not observed in vivo. And what we speculate is that in vitro preparation may be more in this type of non-regulated regime of membrane co-option as opposed to what happens in cells. Also interesting is that it's not just whether it's steep or not. We can put in a hill function. This is quite steep. I think the hill coefficient for this is at 10, and it doesn't do any better. We have to put in a hill coefficient of like 40 or something to get a behavior that looks like in B. And I am aware of no biological process that is authentically modeled with an authentic cooperativity of 40. So we think that this is also kind of not what's going on at this transition. But what might be is if there's a higher order cooperativity that has a power law dependence to ramp up to full activation. And so if we take this type of sloping activation, we can get fits that are pretty good, pretty robust, and actually preferred over what we're starting with. And what we think is that this type of generating function is giving clues about what's occurring at the earliest stages of membrane cooption. And I'm pretty excited about this because that's exactly the part that is virtually inaccessible by experimentation. You can't see these membranes even by electron microscopy until they're fully formed, but we can start putting forth theories about what's happening at those earliest stages that hopefully can give rise to testable experiments. All right, third aim, I just have, I think maybe two or three slides on some of the stress testing that we want to do for these antiviral feedbacks. One comment I have on MAVs, what we would like to do going forward. As I said, we've identified an allele in humans that creates or destroys a cleavage site for the enteroviral proteinases. What I didn't emphasize is that that variant is invariant all throughout other mammals. They always have the site. It's only a fraction of humans that don't have it. In addition, the other site, which I didn't talk about, but is located here, is a splice site insertion that is only in us and old world monkeys. And nowhere else in the genomes do you find these. So these cleavage sites are very human specific, and yet people routinely study coxsackievirus in mice, and they may be missing out on something if they're not working with a human specific, or they're not working with the human allele. So we have gone great pains to teach ourselves how to work with animals and humanize them by adding in human MAVs to a MAVs knockout mouse. And the way that we do this is we use adeno-associated viruses that have a cardiac tropism and are regulated with a cardiac troponin T promoter. And what I'm showing here are a couple of examples, either where we inject these animals with saline or AAV encoding a human MAVs with a self-cleavage site for luciferase, so we can track it by bioluminescent imaging. We inject the animals, we can see bioluminescence, we can confirm it biochemically. And when these animals are sacrificed, we can see luciferase expression in regions of the heart. And where we see the expression of luciferase, we can see expression of the human MAVs. So what we're very excited to do is use these technologies, put in the different MAVs alleles that I described before, infect the animals with coxsackievirus, and then evaluate, does that have an impact on overall outcomes and infectivity in the course of the infection. And we also think that there are other examples of these human-specific susceptibilities that go beyond MAVs. We looked bioinformatically broadly at polymorphisms that occur in greater than 1% of the human population that create or destroy enteroviral proteinase cleavage sites. And we stumbled upon one in the interferon regulatory factor ERF3. This is the key transcription factor that turns on the interferon locus. Here's a crystal structure of it docked to the interferon beta promoter. And there is an arginine in ERF3 that in about 5 to 10% of individuals of African descent has that variant converted over to a glutamine, creating a site. And what we've shown biochemically is that if you purify ERF3 with either the major allele arginine or the minor allele glutamine, and then incubate those immunoprecipitates with recombinant the enteroviral proteinase, we can start seeing glimpses of cleavage products that are consistent with the molecular weight that would be predicted by a cut here. This cut would separate the DNA binding domain of ERF3 from all of its regulatory domains. So we think that it could have a very potent dominant negative effect on cells, and we'll see. But what this opens the possibility is elaborating this abstracted model of MAVs regulation to include additional antagonistic points upstream and downstream to evaluate what really happens when these cleavage sites are active in certain subpopulations of cells, or certain subpopulations of individuals in the broader human population. And then last, what I wanted to touch on in the spirit of antiviral feedbacks is a disclosure that our model in its current form assumes that there is no baseline interferon signaling in the cells, like 0 copies, which is probably not true in reality. What we found is that the models can still foster infection up to about 25 or 50,000 basal copies of interferon stimulated genes. And that seems reasonable, but we don't know how much population-wide variation there is in interferon signaling at baseline in the human population. What we've done is access 3 data sets, 2 which are public, and the other one, since it's European, is basically impossible to get, but we managed to go through the data use agreement to obtain it, of human heart samples, RNA sequenced, either control hearts or individuals that have been diagnosed with dilated cardiomyopathy. And this is a recognized endpoint of individuals with viral myocarditis. So we went after these data sets with the hope that we would be able to pick up instances of human heart tissue with viral infections in them. And in fact, when we have our bioinformatic pipeline realign all the raw sequencing and then search broadly for viruses that are present in the RNA sequencing reads, we can come upon 20 to 30% of those cases where we get detectable reads. Pie charts here showing the distribution of different types of viruses that we see in there. We see a significant increase in viral prevalence in the DCM cases. And what we'd like to use is the presence or absence of cardiopathogenic viruses as a stratifier to look at these transcriptomes and look at the normal ranges of interferon signatures that we see in the broader population. We can plug that into our models and make informed predictions about the breadth of susceptibility to a new enteroviral infection of the heart going forward. I'm finished. I want to acknowledge Andrew Sweat and Cameron Griffiths in my lab, who have been really pushing this forward for the past couple of years now. Paige and Mia were undergraduates who were involved in this in the early stages. They've moved on to better things, and acknowledge current support from multiple private foundations. Thank you.
Thank you very much. I don't know. Normally, we have both talks first and then the discussion.
I'm happy to wait. Yeah, I'll be here. I'm looking forward to the next talk.
The people, okay, if that's okay. Because I was going to say, because in a sense, this is what maybe we should have done as a single talk, because really, but William, are you ready to queue up?
I think so. Can you hear me? You can hear me. That's good. Now the next question is, can you know that bit? The next question is, can you see my screen? You can. Excellent.
Yes.
Okay. Yes. Great. There we go. Actually, I just want to say, Kevin, that was a very interesting talk, and I have it does actually connect us to this in some ways. This operation of swapping out of submodels and the modularity, and it would be interesting to discuss that a little bit more. But I'll just go through this just now. So this talk is based on a paper that we put on the archive not long ago. It's under review. It's together with my co-authors here. I've bolded 2 of the names that are probably going to be familiar to you all. And I'll state right up front the sort of main points of this talk, right? So the first one is, here's an artifact. It's a model. It has a perhaps plausible immune model of adaptive immunity and transmission model in a population. You can get some interesting results out of it, and I'll show you those. But perhaps more importantly, and what I really want to emphasize is the use of language for describing biological processes. So what I want is, I want a language where we can describe what is going on as close as possible to the the concepts and ideas that biologists and immunologists, epidemiologists, all the different ologists are used to working with and like to think of, and to describe the processes in this kind of a language and not have to go once the model's been formulated and then describe them again in a language that is suitable for consumption by a computer. Right? So what I'd like to have is the language support sort of narrative stories about what's going on. It's important that it should be compositional. Compositional means that if there's something else going on, I should be able to just, well, add some more to the narrative and add something and add another story. Or if I don't like one part of the model and I want to swap it with another, I can just remove that and add a replacement sort of story. So this compositional structure of the language is also important. And I'm going to illustrate this by showing an interesting model that that is formulated with this method and has these features. So the starting point for this is some data that was collected from health workers in the UK, the Siren study, and then analyzed by colleagues at the London School, where they looked at what the COVID does in terms of the probability of getting a positive PCR test over time, days since infection, and it sort of looks like this. The gray cloud in the background is the empirical data that they collected from these health workers that were sampled frequently, and the blue curve is fit according to a statistical model. It's good to fit something according to a statistical model, but I would like more of an explanation of what's going on, and so we try to imagine what is the simplest sort of mechanistic model that could reproduce a curve that looks like this. We do this in a rule-based way, so this is a very brief introduction to what rules are. If you think of these as something like chemical reactions, you won't go far wrong. A rule has a left-hand side and a right-hand side, just like a chemical reaction does. However, the left-hand, well, both of them in fact, should be thought of as patterns that can match in some sort of a mixture. In our case here, the mixture is going to be a population. This was originally designed for looking at molecular biology, so you also might imagine these are polymers of some sort, or other such things. The operation of the rule, well, we pick a way that the left-hand side can match in this mixture, and we remove it and replace it with what the right-hand side says. And this is just illustrated in the picture here. The entities that we're dealing with we call agents, but these should not be confused with agent-based models. The different parts of them have names, right? Agents have sites, which are named, say U, that can have an internal state. We make use of this a lot. There can also be bonds between sites, which you might think of as being very useful if you're doing some kind of molecular biology, and less useful in this case, but we still use them a little bit. The agent B on the right-hand side, I've just sort of dropped a bunch of notation just because if I do that, it should be clear. When I do that, it should be clear from context what I'm talking about, and that's just to avoid clutter. At the bottom of the screen, you can see some text, and the text is the representation of this in exactly the same picture, although we've had to write down what the different sites of B are. Exactly the same picture, written down in a machine-readable way. So this is just to emphasize that when we write patterns of agents, and draw them with pictures the way I will be doing, there's a direct correspondence to what you feed to the computer. So there's no need to sort of, no need to mess around with Python or MATLAB or any of those sorts of things. You can write down pretty much exactly what you mean. So, for example, we might have a rule that talks about virus replication. And maybe we don't care about the actual number of virions, we just care within orders of magnitude, so we could track the logarithm of the virion count. And this just says, here's some virus. It's bound to a person, so that site is filled in. So it's a population of virus, a little, population of virus that belongs to a particular person, and it grows. and it grows exponentially. That's all that this says. So that's simple. We have an idea of virus that increases as the virus does. That's what one of the reasons that viruses are problematic. So we'll skip a whole part of the model that is a whole part of not this model. We'll skip a whole part of what could be in a more interesting model that included innate response and all the startup machinery, and we'll go directly to the adaptive response because that's all this model has. Okay, so here's another rule. What do we have here? We have a person, P, and the person has a population of some B cells that have some level of affinity that we call M. And this person also has a population of virus with them. And we imagine that the affinity of the B cells increases according to how much virus there is, right? Which makes sense, right? Because if you have, if you're infected with a virus, and then you've got the dendritic cells coming and grabbing it and bringing it to the germinal centers and presenting it through helper T cells and these sorts of things. This could be made more elaborate, right? This is an example of, here is a simple rule, a simple process that could be replaced with a more complicated one, and we might like to do that for a more detailed model. Here we don't. Here we just say the affinity of the B cells increases according to how much virus there is around, right? And you see the constant of proportionality there in the rate, right? The N times K, the K for affinity maturation. Okay, we can have another thing that happens, right? We have these B cells. What are the B cells going to do? Well, according to their affinity with the virus, according to how well tuned they are to this particular virus, they're going to produce antibodies. And again, nothing more than this. So we have this person. It has a population of B cells that has, some antibodies that come from this population of B cells. And again, we count the antibodies here in logarithmic numbers, right? We just count the logarithm of the count of antibodies. And finally, okay, we have the person. It's got the B cells. Here we don't really care how mature the B cells are, but we do care how many antibodies are around. There are M of them, and these antibodies go, and we imagine that they gum up the virus and enable the virus to be those virions to be collected and eaten by macrophages and whatnot. So according to how many of the antibodies we have, according to our antibody titers, antibody levels that we have there, the population of virus goes down. So here we are with a pretty much very simple. We could replace bits of this with more elaborate rules, and this actually gets on to something that I would be interested to discuss with Kevin, following off of the last talk. But we have a simple model. It does something, and of course, I forgot one rule. Well, if I get down to having no virus or almost no virus, if I have one virion left because these are logarithmic numbers, well, I can have a transition. I can have a rule that says, well, okay, there's no virus, right? This is the clearance, right? So we have a person that's bound to a virus population, and all of a sudden the virus population gets so small that it disappears and the person is no longer infected. So let's make an assumption, right? Say the likelihood of a positive PCR test, imagine it's proportional to the logarithm of viral load. It's a reasonable assumption. It might not be quite right. There's obviously more details, especially according to where you take the sample that you're measuring, for example. But I mean, it's a reasonable baseline assumption. And given this assumption and that model that I just described, okay, we can get quite a nice fit to this data. So the background that we have here is the very same empirical data that came from the papers that I mentioned a few slides ago, and we have the results of the simulation. But we can also do another thing. This is, of course, a stochastic simulation. Each of those, it's not. If you were to take that model, you could, in principle, generate ODEs from it, but they would be extremely high-dimensional ODEs, right? They would be ODEs in something like, as we've done it here, would be something like 8,000 dimensions. It depends on the number of steps you like to have for viral load and antibodies and these sorts of things. So we simulate it stochastically, and because we do this, we get, of course, well, because we do this, we can look at the cross-section, the distribution at each point in time of viral load, and we can do that, and we see this. Right? We see at time 0, we have everybody is infected with a viral load of 1. This is a population of 1,000 or something like this. And we see how the viral load changes in the population according to the time. And what do we get from this? We get that after some initial few days, after time, let's say 9 or 10, something like this, we see a distribution that has a long tail. Right? So, in fact, if you do the sums, you find that by about day 14, about 80% of the viral load in the population as a whole is concentrated in about 20% of individuals. So, in other words, this this process captures the population heterogeneity that we that we in fact see with viral loads in population. So this is nice, and this is interesting that this sort of comes along almost for free, almost automatically from this formulation. So okay, let's make another assumption, right? I mean, we don't want to have well, I don't know. I've been working more in the epidemiology end than the within host kind of end recently. So imagine, this is again an assumption. It's a reasonable one. It's commonly made that the rate of transmission between individuals is proportional to the logarithm of the viral load. So we can make a very natural looking transmission rule, right? We can say here's a person that has some virus, and here's another person with no virus. And then at some rate, you end up with, well, the first person is still there, and the other person now has a little bit of virus, right? They had no viral population, but they became infected, so now they have some. And you can also use the same sort of patterns to look in this mixture of people and their different configurations, and count. You can just define observables. Count observables, count people in different configurations, right? So we have the people with no established immune response and a no viral population, and they're susceptible. And we have the ones that have some viral population, obviously they're infected. And the removed or recovered ones, it's a bit artificial, but we can say, well, these are the ones that have an established immune response but no viral load right now. You don't need to make this distinction in the transmission rule, because suppose that the second P, the blue one there, who's becoming infected, has already an established immune response. Well, they might clear the virus very quickly, right? So, yeah, so that's fine. And you can capture reinfection and so on and so forth with that, without having to have extra rules about that, or to sort of define arbitrary subsections of the population, as you often do in with infectious disease modeling. And if we do this, okay, we can have, here's an epidemic curve. It's a fairly standard epidemic curve, but we can also pick out parts of this curve that will be interesting to look at. So we can pick out the peak. We can pick out a couple of points on the way up and on the way down, and they're chosen basically so that the number of infected individuals are the same on the way up and on the way down. So we have something early in the epidemic on the way up and late in the epidemic on the way down, and we have something sort of in the middle on the way up and in the middle on the way down. Just points chosen just so that they're at the same level. And if we do this, we end up recovering another interesting result, in that when we look at the cross-section of the viral load distribution, we see that the viral load distribution changes according to whether the epidemic is rising or whether it's falling or whether it's stationary, maximum. And this is also interesting because this has been seen in empirical data. It's even been used by, I think, James Hay and colleagues to estimate the current doubling time and reproduction number in the population according to these distributions. But again, this is something that you get that falls out automatically from this simple model. And so that's nice. That's a nice result that comes out of this. Obviously, there are limitations in this, right? I mean, there's no innate immune response. The mechanisms that trigger the adaptive response are absent in this model. Could imagine a model made in the same way with this sort of thing. with all these things in it. The population's unstructured. We use mass action kinetics in this model for how people become infected. We don't necessarily want to do that. What we might like to have is spatial organization, right? We want hierarchical structure. We want spatial partitions. We want my cells are part of my tissues, are part of my body, are part of my body's in my house. I interact with my family in my house. I interact with my colleagues at work, so on and so forth. We want transport between these spatial partitions. But importantly, we want these extra features that do not actually exist in the Kappa language, which is what this is written in. But we do want a well-defined formal language for describing this, again, at the same conceptual level that we want to think of these processes without having to repeat ourselves in a language that is designed to be easy for computers to understand, not easy for humans to understand. And I think that's important, because if we want to make large, scalable models, thinking of these digital twins and that sort of thing, this gap between the conceptual level and the implementation needs to be closed, and that's sort of what I'm attempting to do with this applying these modeling techniques to these kinds of problems. And that's it. Thanks to all sorts of people, and you can get the source code for that. It's very small. It's not on GitHub. It's on SourceHut because I prefer SourceHut. No other reason. Thank you.
Okay, thank you to both speakers. The floor is open. I've created breakout rooms in the case that it looks like we need to have two separate conversations, but I'll create them, but people are welcome to stay in the main room and see how it goes. So please.
I think unless you make us, we who have spoken, go into the breakout rooms, I think it would be funny for people to go there and on their own.
Well, the idea was that I wasn't going to assign people until let's start talking, and if it looks like they're two separate conversations, then we can move people. Usually we have at the beginning of this series, I created breakout rooms every time, and we never used them. But it occurred to me that there might be different kinds of conversation here. People don't seem to. Are people? I just want to make sure that we've got. Okay.
I can start with a question for William if we get the ball.
Sure.
Get the ball rolling.
You two are definitely. If you guys want to talk to each other, that's even better.
Please.
Yeah. Well, the one thing that came to mind, as I was watching your talk, you started with very simple agents and then added increasing levels of complexity. And my question is, where do you start running into limits? For example, if there are so many elaborations off of the agent, won't you run it, even if you simulate a population, won't you have so many combinations that everybody's like a snowflake in the simulation because of just the combinatorial complexity and keeping track of the states? So where does it start becoming computationally problematic with regard to all of the different edges and things in these models?
I suppose there's no simple answer to that. I mean, the more complicated you make the model, the more expensive it is, and potentially the more difficult to interpret it is. This is one of the things that, it's already problematic, because it is quite possible to have an innate model, a model of the innate system, right, that uses interferon and these sorts of things, that will also reproduce that curve. And so now I have, suppose I do that, suppose I write that down. Now I have two models that I can put together and I can have them both happen that sort of explain the same thing. So you can you can very easily, before you even run into computational problems, you can very easily put together a model that it's not really identifiable. But the answer is, there are patterns. I was actually quite careful with the way I formulated this particular one to stay within the sweet spot of the Kappa simulator and what it is good at doing fast. It is easy to write models that will be slow, and sometimes that happens because of limitations in the implementation of the simulator, and sometimes that happens just because you've written something that is really hard.
Okay, other questions? But since you mentioned interferon, maybe come back to Kevin. And my understanding in your model is that the actual mechanism in your simulation has great detail on viral replication, but your interferon-based antiviral resistance is done effectively as a Hill function, and it's perfectly legitimate. But I wanted to ask the question: if you were going to open up the interferon box and start being a little more detailed about your understanding of the interferon pathways, whether it's interferon production, whether it's something like JAK-STAT response, NF-kappa B, where would you start if you wanted to start unpacking that incredible complexity of AMPs and toll-like receptors? And
yes, Jim, I'll answer your question with a comment, and then I'll give you an answer to the question, because it is something that I've gravitated towards, just in terms of modeling philosophy for my group. For mechanistic, we do other types of modeling approaches also, but for like the mechanistic, physical-chemical type of modeling, I feel like we're drawn to either the very detailed, where we think we can capture everything, and we use the model as a sufficiency test to evaluate whether deep mechanistic knowledge can explain what we have, or we go totally abstract if we don't think we're ready. For me, I think we feel very uncomfortable in a middle ground where we have like a faux detail, but it's all phenomenological parameters. I think we lean towards just shouting from the roof that we don't really know everything that's going on at that step, and then we just have it very, very abstracted. That said, where I think that a partial answer to your question is one of the plans that we had, not so much on what happens downstream of the interferon activation when it happens, but the building out of that very simplified MAVS regulatory model to start including things upstream and downstream. In the paper, we didn't do it because we didn't think we need it. And probably for like a normal viral simulation, you don't. But if we start examining variants in the broader population that may be having additional cleavage events occurring upstream or downstream in the same pathway, and there have been reports of double-strand RNA sensors being cleaved. I talked about one of the transcription factors themselves, and we even, in the proposal, we've identified another interferon-stimulated gene that was recently reported to be a restriction factor for coxsackievirus that also has a candidate cleavage site. Now we have to start going upstream and downstream of that. So I think what we may try to do is, rather than have the real detailed one and then go in and add more detail, start with the abstracted one and build the detail as we need. And whether they meet in the middle, I'm not sure, but that's been our approach to it. So I think actually the actual interferon stimulated genes themselves and their mode of action is daunting. Hundreds of genes, all of which seems to do something. And I guess maybe I'll close with a comment. One of the things that's interesting about just interferon stimulated genes is that I actually think it's quite easy to disrupt viral infection to an extent that it has an evolutionary advantage, because there are so many different places that for certain families of viruses are so tuned, optimized over evolutionary time that, and it's an exponential process. So you can have just a minor change in a rate, and over doublings, it can have a real pronounced impact on infection outcomes, and it could be something that would be evolutionarily preserved as an advantage. And so I think that might be, yes, we have all sorts of viruses that we need to be able to combat and so forth, but it actually might not be as difficult as we think to disrupt some of these processes in a twofold, threefold, fourfold way that has a big impact on the overall course of the infection when you look at serial rounds of infectivity.
So I have a question. You used a concept that I like, where you said you can make a mechanistic model that maybe is sufficient to reproduce some data. But one day we might hope to be able to make a necessity claim. We might have to, say that this is actually the right model. Some other model couldn't do it. How does that fit in your worldview?
Well, the grant is on my mind because it's imminent. That is precisely where we're going with that viral replication organelle, where what we currently had is just a workaround, a discontinuous step from no membrane theft by the virus to full-fledged replicating on membranes. And there are very strict requirements for this model to behave about how you make that transition. And so I think that there is actually something very deep by the form that can make that transition. And I think it is some type of multiplicity, cooperative effect that's occurring right at the nucleation of this membrane theft. And in the same way, I think the parallel that I draw, like actin filaments and things, the reason, the early evidence that actin filaments grow as a trimer that nucleated is because the filament initiation goes as actin monomer to the 3rd power. And so we have a relationship that's going to the 6th power. And so I actually think what might be happening is we may have aggregates of nascent polypeptides coming together and getting a 5-member, 6-member group on a membrane. And once you have that, then you're off to the races. But these are our ideas. We actually want to dive into nucleation aggregation theory and on surfaces and figure out if we can get a really principal generating function that gives rise to that change.
Lorenzo, go ahead.
I have one. There's really kind of two that is more, I don't know, high level or philosophical. Starting from what you were saying about the idea of disproving, which is something I have a lot of at heart. This is really something important, I think. And and how do we disprove something that we make on purpose? But then let's say the second part will be how do you feel about what William says in the beginning of his presentation about the semantic of the language that we use? How if we make models in such way that they correspond to the semantic that us biologists use? So when we say, all right, if I do this to these cells, I have upregulation of that gene. This kind of semantic, if is well circumstantiated, can be defined into rules. And sometimes these, if we then use them in specialized or kind of complicate a little bit, we might find a way to say, all right, is that statement correct or not? Or correct as is stated? And this might be enough for the biological progression, because we can do experiments to change a little bit that statement or to do different hypotheses, etc.
Yeah, I mean, from my standpoint, this is, I think, is something that when I've, I don't know if I'm a modeler, biologist, whatever, I can play different roles depending on who I'm talking with. But when I've collaborated with like real molecular cellular biologists, basic scientists, I think one of the things that they've gained the most out of that collaboration is when we start asking about the rules. Because even, Lorenzo, in the example that you gave, to encode that mathematically, if you say, this complex come together, binds and turns on a gene. I'm like, well, does the whole complex, or if you have 2 of the 3 members, can it bind? Or like when this binds, does another factor come off? And a lot of times I never thought about that before. We just draw it as a cartoon, and it all seems to make sense. And we actually have instances in our coxsackieviral model where some of those choices are crucial, where that if you have a complex of polymerases and the viral RNA getting translated and then it docks, like you can't be replicating and translating at exactly the same time, but how does that handoff happen? And how does that relate to the membrane surfaces and things? And so I think that several of those could be framed in qualitative terms. And this is the place where I think that the virologists, infectious disease experts and things, I think they should be drawn to that, because that's some of the places where there are, like, I think, unanswered questions that no one had thought to pose before, because they never had seen the thing fully synthesized, in that way. As we're framing our model as, the synthesis of 70 years of enteroviral research. We're trying to build upon all that hard work, put it together, and if it fits nicely, that should be satisfying. And then we can zoom in where it's not good, and those can be the next places that are worthy of experimentation and testing.
But I think the cartoons are really good. I like cartoons. I think the cartoons are great. I think also, though, that they can be refined just a bit so that they're unambiguous, and so that they actually do encode these details that you're talking about. And from there, transforming that into something that is rigorous enough that can be implemented for a computer automatically without the biologists having to learn to program in MATLAB or whatever, right? I think that's what we need.
Yeah. But this has been attempted. There is a Cell Designer, I think, is a software that does that you draw a diagram, and then you have to put then inside the Hill equation rather than the mass action. This, I agree, is the problem. But my comment on this is that they are beautiful software that people, I guess, have problem of time to actually make properly. Let's say to make a beautiful chart that works perfectly and that it has encoded the mathematical equation inside, and everything is annotated to the papers that actually gave that rule and give confidence on that rule. So if the community grows and attempt to do this, I think this is important to change this cartoon into something more dynamic or more precise. Yeah.
I think the other thing too is it's a little bit of watch what you wish for, because I have one example. This is a totally different project, doesn't involve infectious disease, where we're doing spatial modeling, so partial differential equations on top of signaling, and we're doing it in virtual cell, and things. It was something we were familiar with. We thought it was appropriate for that. And we're working them hard because, like, we want to run that on our supercomputing cluster. We want to be able to import, export data, run simulations in between, and feed them back in. And we are like constantly peppering them with capabilities that they didn't even know that they needed to offer. They've been great and been responsive to us, but it is just like, wow, I didn't even think that, this wasn't geared to run on 1,000 cores, right, to go do the thing, but I guess we could, so we're kind of like putting together bits and pieces. But what started with originally a modeling environment that was supposed to be accessible for cell biologists, it's got kind of, that, but you want to be able to give it the computing power on top of it.
Well, I guess the keep it simple mantra works all the time in every context. But thank you.
Other questions, comments? I think that this issue of falsification, which in a sense both talks address, is interesting. And my first thought was that Kevin, you didn't talk much about falsification, but for example, when you showed that there were certain kinds of transition curves that didn't work, that was I think getting at the kind of falsification that you really wanted to address at the beginning of your talk.
The other example that I'll point out for that was when we were talking about the rhinovirus example. The reason why we sampled through all of these activation energy and enthalpy states is what we really wanted to try to do is figure out if it was impossible to get a model that gave rise to the behavior. Because if it's impossible, we're done. We go do something else, right? And so we were actually trying to kill that hypothesis by giving the model free range to come up with these. And we're still actually trying to, figure out, like we keep talking about in the lab, like, does this project have legs or not? And the way you figure it out is you keep trying to kill it. And if you can't kill it, then you keep working on it.
I mean, classically, models can prove sufficiency, but not necessity. And we run into, biological systems often have backups. So just because your system, the example that William gave, which is you could have two different mechanisms, one which is a population level one, and one of which is an in-host one, which give the same curve. And if you mix them, presumably you get a convolution of two same curves. So if you reparameterize, you can match the data. And so there's really no way to distinguish from that particular set of data. Now, there may be behaviors or parameter dependencies that are different between those two models. But
I guess there's also the question there, though, that I mean, one of the things that I found missing, and this I wanted to bring this up when you were saying, well, okay, can we have this model, and is this model falsifiable? Sort of implying, is this model true, in a certain sense, right? I mean, it gets into that kind of philosophical territory, and it depends. Sometimes it depends on what you want to use it for, right? If you want from what I was doing, if you want like a super simple model of immune response in order to create the population effects that you need to do to study things at the population level, because that's what you're interested in, it doesn't really matter if it's true. It just matters if it's good enough to reproduce that curve, and whether you could reproduce it in another way doesn't matter, right?
Well, but there's a danger there, and I'm going to break in, although Kevin probably is a better answer than I do, which is that if you reproduce one case, you don't know that the situations outside your training set, to use the ML idea, work. And so when you vary parameter X, the variation of that parameter in your arbitrary model might in fact give you something totally different from what varying parameter X would do in reality. And that's why, to me, Kevin's example of showing that he could actually distinguish the temperature dependence of two different viruses was extremely powerful, because that's something that's completely not, as you said, not designed into the model. And it's a stress test of the model that you would never think of, probably, a priori. It's certainly not something that's done in the fitting of the optimization of your parameters. And so to show that kind of dependence emergent from that model, I think is a very, very powerful result. But I don't think there are that many cases that I can think of which have been able to do that kind of generalization. I'm sure there are, and people in the room probably know some, but it's not that common that people do that harsh a test on this kind of model.
No, I agree that the adversarial, that the emergence is super interesting, and I'm not arguing against the adversarial approach at all. I'm just pointing out that I think we do have to, in the thinking about this, we do have to have a what is the model for, right? That's part of it.
Yeah, the thing I was going to add on that, don't take the impression that the way we're proposing to do things is better than all of the other ones, or that we don't use models for the other ways. Like we use them to discriminate hypotheses. We use them because they're useful. We know they're wrong and they know that they're useful. I think the claim that we're making is that there's the space that we're describing is much less occupied. And we're trying to hopefully give examples and show, if you will, other modelers or other teams or whatever, another way to use modeling that is underpopulated. I would argue it's harder to find that way of doing things, but I think there's a lot that modeling could be used in that, with that mindset, that I think could be really powerful.
For sure.
We have a lot of people in the room who normally ask questions, so I'm surprised after these two talks there isn't more. Yeah, I think Kevin, even if we haven't challenged your proposal, I think the approach that you've taken, and I would encourage people to do this, if you're willing to be open about your proposals. I think that using us as a sounding board, maybe people aren't used to doing that, playing that role, so maybe they're quiet for that reason. But I think it's something that this working group could do very effectively. And so the one thing I probably would do is I'd change the order so that we'd have the discussion immediately after the presentation, so that we're thinking primarily of that. But I think it was great experiment. I think this question, I still think this question, it's fascinating. Let's come back. Maybe you mentioned these genotypic variations in particular populations. And of course, evolutionary just so stories are evolutionary just so stories. But those seem like pretty severe sensitivities in those populations. And are there other viruses that use the same kind of blocking mechanisms and the same kind of cleavage approaches, for example? And do you see any do you see a clinical correlates of that observation?
Yeah, so I don't fully know, but I can tell you that SARS-CoV-2 has a 3C-like proteinase that is also glutamine directed. It has a slightly different sequence specificity and things, but it's in that same family, and people try to do protease inhibitors against them. One of the things that's interesting about that topic of doing protease inhibition, that was tried for rhinoviruses actually, and an inhibitor of that same protease didn't make it past phase 2 trials for efficacy. And interestingly, in our models, drugging that protease is very ineffective because the key role that it plays is to mature the polypeptide, which is an intramolecular cleavage event. You're never going to get a drug in that's going to be able to impede that effectively. But if there are at-risk populations that have susceptible alleles, that's a trans reaction. And I don't think anybody would have thought to stratify those drugs based on more or less susceptibility to the protease activity. And so it's a place, we don't have it in the proposal or thing, it's kind of like spitballing at this moment, but the idea that some of these antiviral targeting agents that don't work at the population at large may be effective for these susceptible populations if we can start more susceptible, in essence, host cell genotypes that might be able to reboot those or look at them in a different light. Also, yeah, I don't know, many of these data sets, we build upon, GTEC and all of the GWAS studies and other kinds of things. I don't think that with any type of systems approaches have really intersected greatly up to this point, which is also why we're really kind of diving into it, because it's really straightforward. The consensus sites for these are pretty generic. There's some specificity, and it's not too difficult to find examples where you're predicting cleavage versus not, but you got to figure out if it really happens in cells and does it have an impact. I guess the last thing I'll say, like for the MAVS example that we published, that it has a different, there are different behavior if you're heterozygous for the allele or if you're homozygous for the allele. And for that particular allele, it's really prevalent in the East Asian population. It's about 50% of East Asians have that allele. So 25% on average of East Asians are homozygous for that allele. That's why it comes across. The ERF3 example that I described is much less prevalent, but we think it could serve as a dominant negative. If it gets cut, it could poison the wild type or the major allele ERF3. So some of these you may get partial cleavage, but it's just 50% reduction isn't enough to really tip the system. So this, I think you really need to have the some theory or some position of where that event might happen in a broader infection scheme to make predictions about whether it will be an impactful event or not.
So I have to look back at your diagrams, but this is upstream of interferon response.
Everything is upstream. Yeah, everything is upstream that we've done.
So if you were a susceptible population, then would you expect that people with these alleles would benefit more from, say, interferon treatment than people who didn't have them because they would have naturally lower interferon response to the virus? And therefore, supplementation of the interferon might help. Or would
I would think they would. I suppose that the prediction is that they would benefit disproportionately because what we're predicting is that the virus should be able to squash the endogenous interferon response. And then if it were to be supplemented, and I have drawbacks doing exogenous interferon stimulations and things, but yes, that's where we're operating. And I mentioned really rapidly, we did find one example of an interferon effector gene that appears to have a variant in the population, but it's so poorly characterized at this point. We're quite a bit more drawn towards ERV3 because it's really key upstream of it. Once you get to the interferon response, it fragments out to several hundred genes, and they all have different effects on different viruses and things. So we haven't really quite gone there yet.
Well, but I was thinking precisely because treating with interferon, I guess it hasn't proved as successful as they hoped early on in COVID. One, and two, as you point out, it can have adverse consequences. If you could stratify your population to say that this subgroup is more likely to need it, then and restrict to that subgroup, then it might improve the profile of the drug.
Yeah, I mean, I think, I think the category that I'm going is if we find some of these that have a really strong susceptibilities, is there a diagnostic where you're like, you are an at-risk individual for this family of viruses? And so if it's like that season for that, or there's an outbreak, you should be especially cautious and things. I think that also just having early warning for some of this could have a benefit.
I think you mentioned in your first talk that you did see greater prevalence of some of these viruses in populations which had these susceptible alleles.
There are cyclical outbreaks of Enterovirus in East Asia every 3 years or so. And what we speculate is that it's building up enough naive homozygous individuals in the population to be able to get escape velocity. It's not coxsackievirus B3, but it's other coxsackieviral strains that peak and trough in Japan, China, Southeast Asia.
You mentioned polio. So what do we know historically if there were differences in polio incidence and severity because of that?
I'm like battling around because you can still work with poliovirus. We haven't worked with it yet, but those outbreaks used to happen in the summer. And we haven't done any temperature dependence with the poliovirus model. But we know historically it has a temperature dependence more like CDV3. And we were just talking today with my postdoc about like, once we get this grant out the door, what are we really going to work on when it's pending and things. And we're trying to figure out how do we get this like on firm grounding. And it very well may be as another counterpoint, have the poliovirus delivery module, which is fast. It delivers more like the rhinoviral model, but it doesn't have the experiments in the literature indicate it doesn't have the temperature dependence of rhinovirus. It's more like coxsackie. So I think that could add even more stringency to trying to figure out it actually could provide some clarity about what is so special about the rhinoviral delivery conduit if we're able to further stratify with the poliovirus. We just haven't done that yet.
Thank you. We're almost out of time. Does anyone have a last question for either of our speakers? I know, Ruchira, you've making comments in the chat. Do you have anything you wanted to add?
I guess I could just make a comment that stratification is one of the advantages of mechanistic modeling that you won't get from just, throwing statistical methods at something. It's really mechanism based. So so I felt that this was a good example of that, and maybe something that can be highlighted about the power of mechanistic modeling.
I was going to add one thing that struck me. I'm sorry, I didn't hear that as a question, which is why I broke in. Which was true in both talks, was that one of the critical things was not the goodness of fit of the median or the mean, but what happened to the variance of the results. And that was an important theme in both talks. That doing a fit where you get reproduce the mean or the median is much less interesting than ask the question, what's the real variance in this group of simulations? And Kevin, you talked about how in certain forms of the model you got very wide variance and others you got narrower ones. And William was talking about that as sort of a fundamental characteristic of the approach. And I think that that's something that it's hard enough to fit the mean without trying to fit all of these additional questions about how do you characterize variance. But I think both of you made good points, arguments that we have to address that much more seriously.
And I would say that another area that's out there and available to everyone that I still don't think has been fully utilized are these public data sets. I mean, what we grabbed, we have 1,000 human heart transcriptomes all linked, right? So we can run 1,000 simulations individualized by receptor abundances, interferon signaling state. We can impute the SNPs from the sequencing and things. And each one of these we can run through the models, which in fact what we're proposing to do. And then you get the variance, but you'll also get individual trajectories of patient profiles and figure out, this is what we haven't been able to do up to this point, is the pairing of things. Does high species X correlate with low species Y? You could Gaussian blur everything out and you could show everything. But having that linked data set informed by primary samples that's open and available, that's what all these public repositories are for. I mean, I feel like, gotta leverage to the fullest extent.
Okay, any last comment or question before we close? No, well, I thank both of our speakers for their coming back and being willing to talk again. And I appreciate it. I hope to see how both of these projects develop. Maybe in another 6 months or a year's time, you can give us the next update. Maybe by then you'll have your funding for this project and you'll be telling us about your next approach. Okay, I want to thank everybody. Please send in your ideas for speakers. And we have the digital twin meeting tomorrow morning at 11. Thanks, everyone. Good to see you.
Thanks, Lorenzo. It was a lot of fun. Take care.
Thanks. Bye-bye.